Welcome everyone. I hope you will enjoy this short video where I speak about biophoto cells and algae for a green economy. Over the last couple of decades, the environmental pollution has been one of the major problems affecting humanity. The pollution affecting the Earth is leading to severe climate change that might be soon irreversible. The pollution is mainly caused by an extensive use of the fossil fuels, which are still the basis of our economy and our lifestyle. However, this source of energy, besides causing serious pollution to the environment, will not last by the end of the century. This is not only an economic problem. In fact, it directly affects the health of the world population, and continuing to use the fossil fuel will only exacerbate the environmental and health conditions. One of the possible solutions to this problem is the transition to a green economy combined with a circular economy model. The green economy is a theoretical model of economy development in which the growth is linked to an assessment of the impact that human actions have on the environment. Precisely, it is a form of economy that provides investment in favor of actions that aim to achieve greater energy efficiency, reduce carbon dioxide emissions, and protect the ecosystem. To this end, the use of renewable energy source and the exploitation of the energy efficiency is of fundamental importance. In fact, the main challenge for the present civilization is to design innovative technologies that are capable of addressing the main societal, economical, and environmental challenges, such as globally increasing energy consumption and the necessity to reduce the carbon footprint in the environment to mitigate the climate change. The production of high-efficiency biohybrid photoelectrodes inspired by nature for powering the planet is a highly promising approach for environmentally friendly energy production. The scope is to convert light energy to green chemicals or electricity. Indeed, it has been estimated that the energy captured in 1 hour of sunlight is equivalent to annual global energy produced by human population. But is it possible to use the sunlight for electricity production? It is well known that plants, algae, and some bacteria evolved in order to use the limitless and free sunlight for power their cells. This process is called photosynthesis. The attractiveness of the biophotovoltaic and solar-to-chemical device resides in the potentiality of the photosynthesis process, which was improved all over 3.5 billion years. Understanding how to rationally manipulate the early stages of the natural photosynthesis process to convert sunlight to storable chemical energy in form of fuel and chemicals carries a great potential for developing green technology for solar to chemical conversion with minimal or even negative carbon footprint. For this purpose, the highly efficient natural proteins involved in photosynthetic process, such as photosystem 1 and photosystem 2, are exploited in the semi-artificial photosynthetic system. But let's make a step back and say a few words about the photosynthesis process. Photosynthesis is a process used by plants and other organisms to convert light energy into chemical energy that can later be released to fuel the organism activities. This chemical energy is stored in carbohydrate molecules such as sugar, which are synthesized from carbon dioxide and water. In most cases, oxygen is also released as a waste product. The general equation for photosynthesis is Carbon dioxide plus 2 molecules of water plus photons light energy will give carbohydrate plus molecular oxygen plus water. In plants and algae, photosynthesis takes place in organelles called chloroplasts. A typical plant cell contains about 10 to 100 chloroplasts, mostly in the leaves. The chloroplast is enclosed by a membrane, which is composed of phospholipids inner membrane, phospholipids outer membrane, and an intermembrane space. Enclosed by the membrane is a fluid called stroma. Embedded with the stroma are stacks of thylakoids called grana, which are the sites of photosynthesis. The thylakoids appear as flattened discs, and it is enclosed by the thylakoid membrane, and within the enclosed volume is a lumen or thylakoid space. Embedded in the thylakoid membrane are integral and peripheral membrane protein complexes of the photosynthetic system. In photosynthesis, the light-dependent reaction takes place on thylakoid membranes. The inside of the thylakoid membrane is called, and outside the thylakoid membrane is the stroma, where the light-independent reaction takes place. The thylakoid membrane contains some integral membrane protein complexes that catalyze the light reactions. There are 4 major protein complexes in the thylakoid membrane. Photosystem II, the cytochrome B6F complex, photosystem I, and the ATP synthase. These 4 complexes work together to ultimately create the products ATP and NADPH. Plants absorb light primarily using the pigment chlorophyll. The green part of the light spectrum is not absorbed but is reflected, which is the reason why the most plants have a green color. Besides chlorophyll, plants also use pigments such as carotenes and xanthophylls. Algae also use chlorophyll, but various other pigments are present, such as phycocyanin, carotenes, and xanthophylls. These pigments are embedded in plants and algae in complexes called antenna proteins. In such proteins, the pigments are arranged to work together. Such a combination of proteins is also called a light harvesting complex. In the photosynthesis process, the light-dependent reaction begins in photosystem II. When a chlorophyll A molecule within the reaction center of the photosystem II absorbs a photon, an electron in this molecule attains an excited energy level. Because this state of an electron is very unstable, the electron is transferred from one to another molecule, creating a chain of redox reactions, called an electron transport chain. The electron flow goes from photosystem II to cytochrome B6F to photosystem I. In photosystem I, the electron gets the energy from another photon. The final electron acceptor is NADP. In oxygenic photosynthesis, the first electron donor is water, creating oxygen as a waste product. Photosystem II is extremely complex and highly organized transmembrane structure that contains a water-splitting complex, chlorophylls and carotenoid pigment, a reaction center called P718, pheophytin, a pigment similar to chlorophyll, and 2 quinones. It uses the energy of sunlight to transfer electrons from water to a mobile electron carrier in the membrane, called plastoquinone. The reaction is water, reaction center P718, excited P718, plastoquinone. Plastoquinone, in turn, transfer electron to which feeds them into photosystem 1. The step from where the electron goes from water to the reaction center is performed by a poorly understood step embedded in photosystem 2, called the water splitting complex, or the oxygen evolving complex. It catalyzes a reaction that splits water into electrons, protons, and oxygen. Photosystem 1 complex is an excellent macromolecular nanomachine, able to absorb photons in its photochemical reaction center and convert them into long-lived charge-separated states. The quantum efficiency of the primary change separation in the PS1 complex is 100%. The need to find new sources of energy production for our everyday consumption is one of the most pressing issues of our age. To overcome the dependence of fossil fuels and to access renewable energy resources, innovative paths such as artificial photosynthesis must be followed to go beyond the conventional technologies already available. A different approach to this problem is to take advantage and mimic what nature was able to perfect upon millions of years of evolution. Yet, one of the biggest challenges in modern science is to replicate in an artificial way the most basic processes performed by nature, such as photosynthesis. This fundamental process has been found to be extremely complicated and very difficult to be reproduced in the laboratory settings using artificial, man-made systems or devices. Biohybrid devices are the meeting point of two disciplines that only recently start to interact: biology and material science. The potential of such collaboration is to take advantage of the best of the two fields and build devices that mimic the photosynthetic apparatus of plants in laboratory settings. To do so, at Solar Fuels Laboratory, Center of New Technology, University of Warsaw, we interface protein which can efficiently harvest light upon immobilization on a metallic substrate. This project proposed to use the highly robust photosystem 1 complex from an extremophilic red alga, Cenodoschizon merole, since has been proved to possess remarkable robustness and high activity, a prerequisite for efficient and long-lasting photoconversion in the semi-artificial device. The light absorbed by the photosynthetic protein purified is converted in electron flow toward the electrode, providing the basis for a variety of biotechnological applications, such as solar-to-fuel. We are focused on the minimization of the charge recombination at the interface between the working modules of the device. This is critical to boost the performance of these highly promising solar converting devices. Switching to a green economic model does not only mean use renewable energy sources, but also being able to remedy the pollution already caused. Indeed, another essential challenge of this century is to design an efficient and biocompatible procedure to decontaminate the polluted groundwater and contaminated soil. This involves the study and development of an effective bioremediation system that would allow to detoxify contaminants in the soil and other environments. Bioremediation is defined as the process whereby organic waste can be biologically degraded under controlled conditions to an innocuous state, or to levels below the respective concentration limit as set by the controlling authorities. In other words, bioremediation employs the living organisms, most notably microorganisms, to degrade the pollutants and convert them into less toxic or non-toxic forms. The suitable organisms can be bacteria, fungi, or plants, which have the physiological abilities to degrade, detoxify, or make the contaminants harmless. In some occasions, the organism can be already present on the site or can be isolated from elsewhere and added to the treated materials, using bioreactors as an example. The effectiveness of this method is highly dependent on the applied environmental parameters that influence the microbial growth and the degradation rate. Bioremediation technique depends on having the right microorganism in the right place under the suitable environmental condition in order for the degradation process to occur successfully. The biological process involved in bioremediation technology can be enhanced and focused on the removal of specific hazardous pollutants from soil, water, or atmosphere. It can mineralize waste material into water, carbon dioxide, biomass, or other non-hazardous products, and thus obviates the need for further treatment. Bioremediation can treat a wide range of compounds, apart from municipal waste and processed water. Suitable microorganisms may also be applied for degradation of pesticides, industrial chemicals, components of crude oil, and even compounds that, until recently, were regarded as non-biodegradable, such as chlorinated solvents, chlorofluorocarbons, and other synthetic organic compounds. To this end, in our lab, we are using Cimerola and G-sulfuria, both extremophilic red algae from the Cyanidales family. The order Cyanidales is comprised of asexual unicellular red algae that are known to grow in low pH environment and at moderately high temperature, up to 56 degrees, and are typically found in acidic geothermal habitats throughout the Earth. No other photosynthetic microorganisms are known to inhabit this combination of conditions. The Cyanidales order, since 1981, is thought to consist of three genera: Cynidium, Galdiera, and Cynadioschism. These microalgae, therefore, have the ability to live in very inhospitable environments due to their strong metabolism and can also grow in presence of heavy metals. The study of the detoxification of these algae in presence of heavy metal could lead to a breakthroughs in the treatment of water polluted by these contaminants. Thank you for your attention.